Good morning, dear students.
Good morning, ma'am.
Good morning. Okay, good morning, everyone. So today we are going to start with the new concept of biology to solve a CET questions. That topic is, we know it is a biotechnology principles and processes. Regarding biotechnology, we already discussed in our theory classes. Okay, what is biotechnology? What are the tools of biotechnology? Advantages, disadvantages, examples, etc. Today, we will just brush up certain concepts of biotechnology and then we will solve some questions on it. Biotechnology, principles and processes. Let me brush up certain concepts first, then we will move to the questions. As you can see the slide over here, it is nothing but the definition of biotechnology where we are using application of technology in the field of biology. Why? For the medicine, for the bioremediation, waste management, as well as energy production. So the principles of biotechnology will be genetic engineering. Genetic engineering is defined as introduction of foreign genetic material, the other genetic material, into the host genome. It is called genetic engineering. Tools of genetic engineering are very important in competitive exam. So techniques of genetic engineering or steps is, first one is creation of rDNA, recombinant DNA, gene transfer into host organism. Third one is gene cloning. Sorry. Next. Stanley Cohen and Herbert Boyer, very important. Creation of recombination of DNA was first put forth by Stanley Cohen and Herbert Boyer. Constructed the first recombinant DNA. What they did, they isolated the antibiotic resistance gene from the plasmid of the bacterium Salmonella typhimurium. Okay, Salmonella typhimurium is a bacteria from which plastid, the extracellular DNA which is present in the bacteria. Okay, so that the plasmid, non-genetic DNA, it is called as. Okay, so it is separated from that bacterial cell. Then this piece of DNA carrying antibiotics resistance gene was cut at specific location. So the plasmid carried a gene, okay, which has an antibiotic resistance. It is resistance to the antibodies. So that the gene had been cut at the specific location using the enzyme restriction endonuclease. The popular name of restriction endonuclease is molecular scissors. They are also called as molecular scissors. This all we know. I'm just brushing up the concepts again. Then the next one is gene transfer. The cut piece of DNA was introduced in the plasmid of Escherichia coli. E. coli is a popular bacteria used in biotechnology or genetic engineering as its reproduction rate is very high. Then the piece of DNA was ligated to the vector plasmid by DNA ligase. Ligated means stick. This joining of the two DNA pieces resulted in the creation of recombinant DNA. So they took the plasmid from the Salmonella typhimurium. Then that DNA part, the gene part, has been introduced into the E. coli. The next is gene cloning. Now recombinant DNA is formed. That recombinant DNA was again transferred into the E. coli where multiplication of that recombinant DNA occurs. Then the ability to multiply copies of any template of DNA is called gene cloning. So tools of rDNA technology, you should know: restriction enzyme, cloning vector, DNA polymerase enzyme, DNA ligase enzyme, host organism. Fine. Next, regarding restriction enzymes, we will study later. We will solve first 3 questions now. Be ready to answer. Let us see who will answer first, whether it is YouTube students or the students in the meeting. Fine. Just a minute. So today we will start bit differently. Like we are going to solve from the last question to the first question. Fine. 59th question. How many stages does a bacterial growth curve have? Option A, 5. Option B, 1. Option C, 4. Option D, 3.
Option
C4.
Option C, 4. Janna Jamma Biya. Okay, fine. Prakriti answered as option C. Madhumita answered as option C. Yes. What about YouTube students? Suha TS, Reshma, many of them voted for option C, that is 4. Ramya Nair, great. Okay, fine. Please keep up the same spirit until the end of the session. First 5 questions answer ಮಾಡ್ತೀರಾ ಆಮೇಲೆ ಬಿಟ್ಬಿಡ್ತೀರಾ. So don't do that. Okay. So let us see what is the answer for it. 1 minute. Yeah. Option number C, that is 4. The number of stages is 4. So here the number of phases are 4 and the name of the phases are lag phase, log phase, stationary phase, death phase. Lag, log, stationary, death phase. 4 phases are there. 58th question. 57th question. Culturing method, dash culturing method produces higher biomass and higher yield of the desired product. Which culturing method? We have different types of culturing method, right? Okay, in vitro methods. So, ಯಾವ method ಅಲ್ಲಿ ಜಾಸ್ತಿ ಬಯೋಮಾಸ್ ಮತ್ತೆ ಜಾಸ್ತಿ ಪ್ರಾಡಕ್ಟ್ ಸಿಗತ್ತೆ ನಮಗೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಆಪ್ಷನ್ ಎ ಬ್ಯಾಚ್, ಆಪ್ಷನ್ ಬಿ ಕಂಟಿನ್ಯೂಸ್, ಆಪ್ಷನ್ ಸಿ ಮಿಕ್ಸ್ಡ್, ಆಪ್ಷನ್ ಡಿ ಹೈಬ್ರಿಡ್.
Option B, continuous.
Option B, continuous.
Option B, continuous. Kanchana Raj answered as option B. That answers also voted for option B. And some of them voted as option D. Okay. Fine. Ramya, Prakriti, Madhumita, Lohit. Okay, Soumya Narayan. So here, let us see what is the answer. Dash culturing method produces higher biomass. Shashi also voted for option B. What about YouTube students? Option B. Fine. The right answer is option B, that is the continuous culture method produces larger biomass leading to higher yield of the desired protein. Fine. Now, just a minute. Okay. In which DNA sequence does the restriction enzyme E. coli always cleave the nitrogen sequence? Okay. In which DNA sequence does the restriction enzyme ಇದೆ ಹೌದಾ? So E. coli ಯಾವ ನೈಟ್ರೋಜನ್ ಸೀಕ್ವೆನ್ಸ್ ಅಲ್ಲಿ ಈ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ ಕ್ಲೀವ್ ಮಾಡತ್ತೆ ಅಂತ. ಆಪ್ಷನ್ ಎ AAGCET, ಆಪ್ಷನ್ ಬಿ GGATCC, option C GAATTC, option D TGGCA. Lohit answered as option C. Chandru, option C from the chat answers. Prakriti, Soumya Naral. Yes, what about others? Be fast. Ramya and Suhati, yes, voted for option C. Great.
C, ma'am.
Okay, Kanchana Raj told as option C. Madhumita and Shashi answered as option C. What about YouTube students? Yes. Be fast. Fine. So the right answer is option C, that is GAATTC. GAATTC. All of you attempted and your attempt is correct. Now, the next question is 31st question goes like this. T plasmid used in genetic engineering was modified by. T plasmid means is a type of a plasmid. ಅದನ್ನ modify ಮಾಡ್ತಾರೆ. ಹೇಗೆ ಮಾಡಿಫೈ ಮಾಡ್ತಾರೆ, ಅದನ್ನ ಆಲ್ಟರ್ ಮಾಡ್ತಾರೆ, ಅದನ್ನ ಚೇಂಜ್ ಮಾಡ್ತಾರೆ, ಯಾವ ರೀತಿ ಅದನ್ನ ಚೇಂಜ್ ಮಾಡ್ತಾರೆ ಅನ್ನೋದನ್ನ ಕೇಳಿದ್ದಾರೆ. ಆಪ್ಷನ್ ಎ ಆಡಿಂಗ್ ಟ್ಯೂಮರ್ ಫಾರ್ಮಿಂಗ್ ಜೀನ್ಸ್, ಆಪ್ಷನ್ ಬಿ ಡಿಲೀಟಿಂಗ್ ಟ್ಯೂಮರ್ ಫಾರ್ಮಿಂಗ್ ಜೀನ್ಸ್, ಆಪ್ಷನ್ ಸಿ ಆಡಿಂಗ್ ಜೀನ್ಸ್ ಫಾರ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯೇಸಸ್, ಆಪ್ಷನ್ ಡಿ ಡಿಲೀಟಿಂಗ್ ಜೀನ್ಸ್ for endonucleases. Okay, I got two answers. Prakriti, Lohit.
Option B, ma'am, deleting tumor forming gene.
Okay, Kanchana Raj voted for option B. Reshma, Suhati, yes. Deepti, great. Madhumita, on answer, everyone answer honestly. Simply don't copy the previous answers. You're cheating for yourself. I'm not blaming anyone specifically. I'm just suggesting you don't copy the answers of the previous messages. Okay, most of you voted for option B. Let us see what is the correct answer. Read option B, that is the role of T plasmid in biotechnology is that it may be converted into a cloning vector by deleting the pathogenicity gene, the genes which causes pathogenicity. Okay, the T plasmid delivers a DNA fragment into normal plant cells, causing them to convert into tumor cells. So option B is the right answer. Now let us proceed with certain concepts again. Okay, so we were discussing regarding restriction enzymes. I want any one of you to voluntarily read out the slide. Who wants to read?
Restriction enzyme. Restriction enzyme belongs to a class of enzyme called nucleus. These are of two types, exonucleus and endonucleus. Exonucleus cut the DNA at the ends, where the endonucleus make the cut at specific point in the DNA.
Very good. So in restriction enzyme, we have two types, exonucleases, endonucleases. Exo, out, which cut the DNA at the ends, endo, in, which cut the DNA at the specific point in the DNA. Next we have, regarding restriction enzyme, we have certain important points which we need to remember. First restriction, endonuclease isolated from hind two. It was isolated from the bacterium Haemophilus influenzae. Please do remember that it is isolated from the bacterium Haemophilus influenzae. Today we know more than 900 restriction enzyme isolated from 230 strains of bacteria. Fine. Restriction, this one, which we already discussed now. Then palindromic sequence. What do you mean by palindromic sequence, guys? Palindromes are the group of letters which read same both forward and backward. Malayalam, Malayalam. So like that only we have palindromic sequence as a nucleotides. Okay, GAATTC. When you reverse it, CTTAAG. 5' to 3', 3' to 5'. So a palindromic sequence is a sequence which reads the same on the two strands of DNA when orientation is kept same. See, GAATTC, GAATTC. Now, so cutting, pasting, restriction enzymes are there which are called as molecular scissors. Type 1, 3 types are there. Type 1, type 2, type 3. The ends. Fine. Now let us discuss some questions again. Be fast, be active. Okay. Next question. Yeah. The Human Genome Project, HGP, is closely tied to the rapid development of a new area of biology, which is called as option A, biotechnology, option B, bioinformatics, option C, biogeography, option D, bioscience. Who is going to answer first?
Option B, ma'am, bioinformatics.
Okay. Ravi Kumar, option A, Chandru, option B, Suhati, yes, option B, Ramya, option A. So there is a fight between option A and option B. Padmayan, Saumanara voted for option D. Okay, Kanchana Raj told or voted for option B? Fine. What about others? Be fast. Some of you will not respond only. By Shashi, Reshma. Answer for or respond to every question, please. Okay. Now, let us see the answer. Option B, that is bioinformatics, is a right answer. Some of them got confused with the biotechnology. So, Human Genome Project is related to bioinformatics. 33rd question goes like this, guys. Who discovered polymerase chain reaction technology? Option A, Carey Mullis. Option B, Saiki et al. Option C, Craig Venter. Option D, Maxam Gilbert. Maxam and Gilbert. Okay, I got the first message by Lohit. He voted for option A. What about others? Be fast. YouTube students, Iti N M option E, Padma N, Suha TS, Prakriti, Reshma. Okay, great. Yes.
Option A, ma'am.
Option A, Carey Mullis. Okay. What about others? One alphabet type madak estotrappa. Here you don't have any OMR, anything. No one will scold you if you write or if you answered wrong. Okay. Fine. Most of you answered as option A, that is Carey Mullis. Option A is the right answer. 34th question goes like this about gene gun method. We all are aware of gene gun method. So there are certain statements which are given. Out of this statement, which of the statement given above are correct? Out of 4 statements, which statements are correct related to gene gun method? Option statement 1, this method is also known as ballistic technique. Statement 2, in this method, cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA in plants. Third statement, important crops like maize, rice, wheat have now been transformed by this method. Which of the statement given above are correct related to gene gun method? I got only one message. Padma N, option B.
Option B, ma'am.
Okay. Option B by Kanchana Raj. Fine. Just a minute. Option A by Soumya Nara, Ramya Nayak. Option B.
Ma'am, screen share ಮಾಡಿ ಮತ್ತೆ ಅಮ್ಮ ಹೋಸ್ಟ್ ಮಾಡಿ.
Okay, most of you answered as option A. Some of them voted for option B. Fine. Okay, Kanchana Raj, you have doubt between A and B. First and second, first and third. Okay. About gene gun method. Let us see. Anyone answered it as? Okay, most of the answers are related to option A. Okay, fine. Option D is the right answer. All 3 statements are right, guys. Gene gun method andre eno adike ballistic technique antanu kuda karitivi. Alli en ಮಾಡ್ತಾರೆ high velocity micro particles gold ಮತ್ತೆ tungsten na bombard ಮಾಡಿದಾಗ with the DNA in plants. ಈ ಮೆಥಡ್ ಅಲ್ಲಿ ಇದನ್ನು ಶೂಟ್ ಮಾಡಿದಾಗ bombard ಮಾಡಿದಾಗ ಅದಕ್ಕೆ gene gun method ಅನ್ನೋ ಹೆಸರು ಬರುತ್ತೆ. So important crop plants like maize, rice and wheat are now have been implemented by this method. So all 3 statements related to gene gun method are right. Option D is the right answer. And I did not get any right answer. for this question actually. It's okay. Panchanaraj, don't, no need to worry about A and B. The answer is option D. Fine. Let us brush up certain concepts again. Okay. So here is, what do restriction enzymes help us to achieve? Means what is the use of restriction enzymes? They help in generating DNA fragments with precise ends. DNA fragments in now ಮಾಡಬಹುದು. These precisely generated ends can be pasted and it can be separated. It can be pasted to the vector which we want. The process of pasting is called ligation. It requires an enzyme called DNA ligase. The product after ligation is called recombinant DNA molecule, or it is also called as clone. Now, the next concept is, what are cloning vectors? Cloning vectors andre enu anta. DNA molecule that carries foreign DNA into a host cell. Cloning vector andre ಅದು ಒಂದು DNA molecule ಆಗಿರುತ್ತೆ. ಅದರ ಕೆಲಸ ಏನು? It takes the foreign DNA into the host cell. Host cell ಅಲ್ಲಿ it replicates along with the foreign DNA and produces many copies of itself as well as foreign DNA. So this is called cloning vector. Okay. We have the cloning vector POC 19 here. Classmates or bacteriophages. Vectors. Okay. Vectors means DNA molecules capable of accepting foreign or new DNA are called vectors. Most common are plasmids. We know that. Then plasmids will be circular extrachromosomal pieces of DNA. These carry antibiotic resistance gene. This is a structure of plasmid. CDR, then restriction enzyme site, ORI, restriction enzyme site, C, selectable marker. See the position of selectable marker, bacterial DNA, and these are plasmids. These all we studied in the theory classes. Then vectors for cloning genes in plant. Agrobacterium tumefaciens can cause the plant disease which is called crown gall by transferring specific genes to the dicot plant. Dicot plant, Agrobacterium tumefaciens. So iden madutte, it contains a large plasmid which is called as T plasmid. So T plasmid is found in bacterium, Agrobacterium tumefaciens, which can deliver tDNA to transform normal cell into a tumor cells. Plant genetic engineers have used this natural transformation system as a vehicle for the introduction of foreign DNA into these plants. Okay. Now let us discuss certain questions again. Just a minute. Okay, 35th question. Let us focus on questions now. A cloning vector, just now we studied regarding this, guys. A cloning vector has two antibiotic resistance genes for tetracycline and ampicillin. A foreign DNA was inserted into the tetracycline gene. Non-recombinants would survive on the medium containing. In kelidare andre cloning vector ide, adakke two antibiotic resistance genes ive. That is for tetracycline matte for ampicillin. Ee time alli a foreign DNA was inserted. Foreign DNA na in martivi navu cloning vector ge insert martivi. Yakade into the tetracycline gene. Avaga non-recombinants would survive on the medium containing. Tetracycline hatra foreign DNA ನಾವು insert ಮಾಡಿದಾಗ ಅಲ್ಲಿ recombinant DNA formation ಆಗುತ್ತೆ. ಎಲ್ಲಿ recombinant DNA formation ಆಗೋದಿಲ್ವೋ ಅವು ಯಾವ ಮೀಡಿಯಂ ಅಲ್ಲಿ ಸರ್ವೈವ್ ಆಗ್ತವೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. Option A, ampicillin but not tetracycline. Option B, that is option 2, tetracycline but not ampicillin. Option 3, both tetracycline and ampicillin. Option 4, neither tetracycline and ampicillin. Yes, what is the answer? Think over it and then answer. Read the question properly, think over it. Chandru answered as option 3. both tetracycline and ampicillin. Lohit answered as 3 or 4. You can't answer like this, Lohit. In the OMR, you can't mark as 3 and 4, right? Kanchana Raj, option C.
Option
Parish?
C, ma'am. Option 3.
Option 3. Okay. Reshma, option 3. Prakruti, option 4. Suhati, yes, option 3. Okay. What about others? Ramya M. Nayak, option 3. What about YouTube students? Fine. Okay. Let us see. The right answer is option number 3. That is both tetracycline and ampicillin. So what is that? Prakruti, can you read out the slide, please? Are you here? Just switch on your mic and read out the slide. No response. Great. Reshma, can you read out the slide? Fine. ಯಾರಾದ್ರೂ ಓದಿದ್ರೆ ಬೈತಾರ ಬಂದು. ಸೌಮ್ಯನರ, can you read out the slide?
Yes, ma'am.
Yeah.
The DNA molecule PBR322 was the first artificial cloning vector constructed by the Bolivar and Rodriguez. It has 2 antibiotic resistance genes, ampicillin resistance gene and tetracycline resistance gene.
Okay. So the non-recombinant DNA recombinants will survive on both because the cloning vector has both tetracycline and ampicillin. So the right answer is option number 3. Here we have to remember that artificial cloning vector PBR322 is discovered by Bolivar and Rodriguez. Okay. Next question goes like this, guys. Identify a character that is not desirable in a cloning vector. ಕೆಳಗಡೆ ಕೆಲವೊಂದಿಷ್ಟು ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ನ ಕೊಟ್ಟಿದ್ದಾರೆ. ಈ ಕ್ಯಾರೆಕ್ಟರ್ ಅಲ್ಲಿ ಯಾವುದು ಕ್ಲೋನಿಂಗ್ ವೆಕ್ಟರ್ ಗೆ ರಿಲೇಟ್ ಆಗಲ್ಲ. It is not related to cloning vector. Option 1, an inactive promoter. Option 2, an origin of replication site. Option 3, selectable markers such as genes for antibiotic resistance. Option 4, one or more unit restriction endonuclease sites.
Option 1, ma'am.
Option 1. Shivu Prashant, option A. Soumya, option A. Thank you, Soumya, for the quick response and reading the slide. Padmayan, option A. Kanchana Raj, option A. That is option 1. Prakriti, option 4. Deepti, option 1. Samrin Taj, option 1. Yes, what about others? Be fast. Okay. Let us cross-check the answer. The right answer is option 1. That is an inactive promoter is not a character of a desirable cloning vector. Here you can see the characteristics of the desirable cloning vector. Who wants to read? Yes, anyone?
The following are the features that are required to facilitate cloning vector into vector. First one, the origin of replication. This is a sequence from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cell. Selectable marker. In addition to or origination, the vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permeating the growth of transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacteria. And the third one is cloning site. In order to link the alien DNA, the vector needs to have a few, preferably single recognition site for commonly used restriction enzyme.
Thank you. Thank you, Kanchana Raj. Okay. So here, all an origin of replication site, it is there as a feature or a characteristic. Then selectable marker, it is there as a feature or characteristic. Then cloning site, that is one or more unique restriction endonucleosides and the cloning sites. So it is also a feature of the cloning vector. So which is not a feature? Option 1, that is an inactive promoter is the answer. Good. The next one is the enzyme used in the polymerase chain reaction. Very easy question. Option 1, DNA dependent RNA polymerase. Option 2, RNA dependent DNA polymerase. Option 3, DNA dependent DNA polymerase. Option 4, RNA dependent RNA polymerase. Got only one answer. Padma Yan answered quickly and she voted for option A. What about others? Enzyme used in polymerase chain reaction. PCR alli yava enzymes anna use ಮಾಡ್ತಾರೆ. Very easy question. Why there are no answers? ತುಂಬಾ easy question ಅಂತ answer ಇಲ್ವಾ? Lohit option A, DPM option A. What about
Option
others?
3.
Option
3.
Option 3. Kanchanaraj voted for option 3. Samrintaj and Padmayan again voted for option 1. Yes. Fine. Okay. The enzyme used in polymerase chain reaction. Reshma also answered. The right answer is option 3, that is DNA dependent DNA polymerase. Fine. Okay. So what is DNA polymerase? That we have to read. Now, just a minute. Ramya Nayak, can you read out the slide? It is hardly 2 to 3 sentences. Ramya Nayak. Samrin Taj.
DNA polymerase are a group of enzymes that catalyze the synthesis of DNA during replication. The main function of DNA polymerases is to duplicate the DNA contact of a cell during cell division. They do so by adding nucleotides at 3-OH group of growing DNA strand.
Thank you. So this is regarding DNA polymerase. Okay, DNA dependent DNA polymerase. Next question. Calcium is a dash cation. Okay, monovalent, trivalent, divalent, tetravalent.
Option C, ma'am, divalent.
Okay, option C, divalent. What about others? That answers are also voted for option C, that is divalent. Okay. Option C is the right answer. The next question goes like this. Which metal microparticles are used in gene gun? Option A, aurum. Option B, nitrogen. Option C, magnesium. Option D, cupra.
Option A, aurum.
Option A, aurum. Deepti answered through a mic. And chat answers I got as option A by
Option A.
Suha TS, Deeptian, Kanchana Raj, Lokit, Soumya Naral, and Samrin Taj. Okay. What about others? Why others are not answering? Easy question ಕೊಡ್ತಿದ್ದೀರಾ ಅಂತ ಹೇಳ್ತೀರಾ. Easy question ನೀವು ಆನ್ಸರ್ ಮಾಡಲಿಲ್ಲ ಅಂತಂದ್ರೆ ಇನ್ ಟಫ್ ಕ್ವಶನ್ಸ್ ನೀವೇನ್ ಆನ್ಸರ್ ಮಾಡ್ತೀರಾ? Attempt easy one. Just attempt at least. Okay, Ramya, option A. Option A is the right answer. Aurum, it is also called as gold. Which method uses disarmed pathogen vector? Disarmed pathogen vector anna yava method alli use ಮಾಡ್ತೇವೆ? Option A, transformation. Option B, biolistics. Option C, insertional inactivation. Option D, sequencing.
Option B, ma'am.
Option B. Kanchana Raj told as option B. That answers I got as Reshma, by Reshma, Samrin Taj, Deepti, Lohit, Suha. Okay. Few of them voted as option A, Padma. Okay. Most of them voted as option B, that is biolistics. Ramya Nayak. Okay, fine. Let us check the answer. Option B is the right answer. So here, biolistics or gene gun method is a method which uses disarmed pathogen. A pathogen from which virulence or disease-causing genes will be removed. It is called as disarmed pathogen. It is a process in which DNA is transferred into a host without any risk of infection. So this method is used, this procedure is used in the biolistics method. Fine. Next. The nucleic acid in most of the organism, very easy question. Option A, DNA, option B, RNA, option C, proteins, option D, lipids.
Option A, ma'am.
DNA, ma'am.
Option A.
Option
Okay,
A, ma'am.
great. Chandru and Lohit answered through chat. Padmayan also voted for option A. Very easy question. Option A, that is DNA, is the right answer. Bacterial cells can be lysed by using dash enzyme. Bacterial cells ಅನ್ನ ಲೈಸ್ ಮಾಡೋದು ಹೇಗೆ? ಯಾವ ಎಂಜೈಮ್ ಮೂಲಕ ಅದನ್ನ ಲೈಸ್ ಮಾಡ್ತೀವಿ ಅಥವಾ ಕಟ್ ಮಾಡ್ತೇವೆ? Option A, protease. Option B, lysozyme. Option C, lipase. Option D, cellulase. Yes. Option C, Padmayan, and some of them answered as option B. Okay. What about others? Chandru, option B. Okay. Soumya, option B. The right answer is option B, that is lysozyme. The fungal cells can be lysed by using dash enzyme. Option A, lysozyme. Option B, cellulase. Option C, chitinase. Option D, lipase.
Option C, ma'am.
Okay, Soumya answered as option C. Lohit answered as option C through the chat. Kanchanaraj also voted for option C. What about chat answers? Some of them have confusion, that is, it is related to lysozyme. Option A. Fungi-alli. They are not asking. They are asking in fungi. Okay. Option C, that is chitinase, as the cell wall of fungi is made up of chitin. So chitinase is the lysing enzyme used to open up the fungal cells. Then 45th question goes like this, guys. The enzyme which cleaves DNA, ligase, lipase, DNAs, RNAs. DNA na cleave mado enzyme yaudu anta kelidare. Okay. That answers I got. What about the others?
C, ma'am.
Okay. Sorry. Kanchanaraj also voted for option C. Yes. Shivu Prashant, Deepti M. Okay, ligase alla. Ligase is used to stick the ends. It is not ligase. Okay. The right answer is option C, that is DNAs. Okay, it catalyzes and breaks down the phosphodiester linkages of the DNA. It is a type of endonuclease. Ligases are the enzyme used in joining of two strands. So the answer is DNAs. 47th question, which macromolecules are present along with DNA within the cell? DNA bittu matte yava macromolecule irate? Viruses, bacteria, vectors and organelle, lipids and polysaccharides.
Option D, ma'am. Lipids and polysaccharides.
Good. Option D. I got chant answers as A, D, D. Most of them are voting for D. Okay. What about others? Suha, Padmayan, Soumya Naran, Chandru, Lohit answered in chat. What about others? Reshma. Most of them are, they will neither answer through a chat. They will neither answer nor answer through a mic. Then you guys are not utilizing the class properly, I think. Okay, fine. Let us discuss the answer here. Viruses, bacteria, vectors, and organelle. These are not related to the macromolecules. Okay. So that is why the right answer is lipids and polysaccharides. Option D is the right answer. 48th question goes like this. Dash is a monomer of lipid. amino acid, monosaccharide, fatty acid, and sugar. Okay.
Option B, ma'am.
Option B. Okay. Chat answers I got as B and some of them confused with option C. Fine. Monosaccharide. Okay. Monomer of lipid. Monomer, single unit of lipid. So here amino acid is a monomer of protein. Monosaccharide is a monomer of carbohydrate. Sugar is an example of a carbohydrate. Fatty acid is a monomer of lipid. So the right answer is option C, fatty acids. Fine. Dash is the monomer of proteins. Option A, monosaccharide. Option B, fatty acids. Option C, amino acid. Option D, glycerol.
Option C, amino acid, ma'am.
Okay, option C, amino acid. And the chat answers are option C. Lohit and Chandru answered to chat. Ramya M. Nayak, Suha TS. Yes, what about others? Reshma. Fine. Okay, Padmayan. The right answer is option C, that is amino acid. Amino acid is the right answer. Okay, 50th question. Dash is the building block of carbohydrates. Option A, monosaccharide. Option B, amino acid. Option C, glycerol. Option D, fatty acid.
Monosaccharides.
Sorry, Deepthi answered as monosaccharides.
Option A,
That answer-
monosaccharides, ma'am.
Okay, Kanchana Raj voted for option A. Ramya, Suha, Saumya, Naral. What about YouTube students? Yes. Okay, most of you answered it rightly. That is, the correct answer is monosaccharide. Amino acid is the building block of protein. Glycerol is a product of lipid. Fatty acid, it is also a building block of lipid. Then, option A is the right answer. The enzyme which cleaves RNA is called DNase, ribonuclease, ligase, and protease. RNA ಅನ್ನ cleave ಮಾಡುವಂತ enzyme ಯಾವುದು?
Option B, ma'am.
Option B, ma'am.
Option B. Okay. Padma answered as option B. Kanchana Raj also voted for option B. And majority of the chat answered by Suha, Ramya, then Padma and Chandru. Most of the answers are also voted for option B. Let us check what is the answer. Option B is the right answer regarding ribonuclease. Okay, Deepthi, can you read out the slide? Here, Deepthi M. No. Okay, Padmayan, can you read out the slide?
Ribonuclease is the enzyme responsible for catalyzing the cleavage of RNA. RNAs are a class of hydrolytic enzymes that catalyzes both the in vivo and in vitro degradation of ribonucleic acid molecules into smaller components. The nucleus operates at the level of transcription and translation and breaks down the RNA by cleaving the phosphorus oxygen bonds.
Great. So the RNAs or ribonucleases cleaves phosphorus and oxygen bonds. Option B is the right answer. 52nd question. The antibiotic resistance gene can be used as dash marker for selecting transformants. Antibiotic resistance
Option A, ma'am.
gene ಆಗಿ use ಮಾಡ್ತಾರೆ. Option A selectable, option B differential, option C similar, option D radioactive.
Selectable.
Complete
Option
the
A,
options. Yeah, I appreciate the way of answering by you guys, but until I complete the options, please don't say the answer. Okay, that answers are answered by Deepthi, Ramya. Okay, only two. Lohit, Chandru, Sagar. Okay. The right answer is option A, that is 1 minute. Okay, the right answer is option A, that is selectable. It's not D. 53rd question, the common bacteria used in genetic engineering is the common bacteria. Option
B,
A,
ma'am.
S typhi.
Option B.
Okay. Until I complete reading all the options, please don't answer through mic. Option A, S typhi. Option B, E. coli. Option C, rhizobium. Option D, yes or yes. I got the chat answers as option B, and Deepthi and Padma also answered as option B, that is E. coli is a right answer. Why? Because it is widely used. It has a generation time of about 20 minutes. Generation time. The multiplication rate is very fast. Next, rDNA technology is associated with which scientist? Christian Graham, C. Darwin, Robert Koch, Herbert Boyer. Okay, Lohit answered in a chat box. What about others? Sagar also voted for option B.
Option D, ma'am.
Option D, okay. Deepti M, option D. Suhati, yes. Soumya Naral, option D. Okay, most of them are
Option
saying
D,
it is associated, okay, with Herbert Boyer. Let me check the answer. The right answer is option D. That is, Boyer and Cohen's achievement represented an advance upon the ingenious techniques developed by Paul Berg. So, Paul Berg and then by Boyer and Cohen's for inserting viral DNA into a bacterial DNA. Next. Okay. Which culture system is used to obtain cells in the exponential phase? Exponential phase alli cultures cells na ನಾವು obtain ಮಾಡಬೇಕಾದ್ರೆ ಯಾವ culture system ಅನ್ನ use ಮಾಡ್ತೇವೆ? Option A single, option B monoclonus, option C continuous, option D batch. Yes. Okay. Most of them answered or voted for option C, that is continuous, right?
Option C, ma'am.
Option C, continuous cultural system. Option C, here, why they will use continuous cultural system? Because their nutrient availability will be unlimited. So the cells are maintained in a continuous exponential phase. That is why continuous culture medium will be used. 57th question. Dash culturing method produces higher biomass. Just now we discussed it. Higher yield of the desired product. Yes.
Option B, ma'am. Continuous.
Option B continuous. Chat answers are also voted for option B. Okay. So the right answer is option B. The right answer is option B. This we discussed already. Just a minute. Now we will brush up some concepts again. See, tumescence gall, disease or the disorder caused by Agrobacterium tumescens. It is a soil bacteria which causes common plant tumors in the plants, which is also called as crown gall disease. The genome of Agrobacterium tumescens C58 has been sequenced completely. So that genome is sequenced completely and consists of circular chromosome and a linear chromosome, circular, linear, and two plasmids. Then T plasmid, this is a tumor producing plasmid, left border, auxin, cytokinin, right border. Then, okay, so this is a healthy plant. This is a wound in a stem. When the plant gets wound, Agrobacterium enters into it, which undergoes rapid cell division and causes crown gall disease to the plant. Then, okay. Fine. the main important point. Retroviruses in animals have the ability to transform normal cells into cancerous cells, retroviruses. Then retroviruses will be disarmed, means the pathogenicity gene will be removed from them, and now we will use it to deliver desirable gene into the animal cell. Retroviruses, okay? Then gene gun, disarmed pathogen, micro injection, gene gun technique. Yeah, this is a gene gun technique. DNA coated with golden particles will be shooted, and this is a plant cell's DNA. Okay, now it has been entered, like foreign DNA has been entered. Now a plant cell with a new gene will replicate and the cell division occurs. Then we will get a transgenic plant or genetically modified plant. Process of recombinant DNA technology: isolation, cutting of DNA, amplification, insertion, obtaining foreign gene product. So these are the steps. See here, this is a plasmid, DNA with contents, cut ends E. coli at arrows, this one and this one, colored region. Now these are isolated and then these are inserted into the plasmid. Then construction of recombinant DNA using a vector. Next. See, DNA containing desired gene will be removed from the cell. Then enzymes cut out desired gene. Then the genes or the fragments of DNA will be inserted in the plasmid. Then the plasmid is inserted into the bacteria where multiplication occurs. Bacteria reproduce, creating a large number of bacteria with new characteristics. Again, the plasmid is derived. Then we will get the gene of interest. This is a rDNA technology. Then polymerase chain reaction, very important reaction. In this, primers are there which are called small chemically synthesized oligonucleotides. These are oligonucleotides which are complementary to the small DNA or regions of DNA. Then we have Taq polymerase, TAQ, thermostable DNA polymerase which has been isolated from Thermus aquaticus. Taq polymerase is isolated from Thermus aquaticus, a heat-loving bacterium. Thermus aquaticus is a heat-loving bacterium which is found in hot springs of Yellowstone National Park. Then nucleotide bases. These are main processes or the terminologies. Denaturation andre, DNA is heated to cause its two linked strands to separate. That is called denaturation. Annealing andre, the temperature of the mixture is lowered. Mixture the temperature low mardaga, the primers started pieces of DNA bind to the separated DNA. For that we have to lower the temperature. For that process we will call it as annealing. The next one is extension of polymerization. The Taq polymerase enzymes is there, no? It extends the primer using the nucleotides, complement base pairing, and copy the DNA rapidly. This is how polymerase chain reaction occurs. Application: to understand molecular events, it can be used to make precise gene maps. Useful chemical compounds will be produced in the diagnosis of diseases. So, idana now biotechnology applications chapter alli in detail agi kalite. Fine. Okay. So this is regarding a small concept, that is biotechnology principles and processes. It is a small concept, so we will get around 3 marks from this chapter. 3 marks. Okay. Any doubt?
No, ma'am.
Okay. Then let me end up the session. Tomorrow's topic is biotechnology's application. Okay. So from that also we will get around 2 to 3 marks. Be prepared. Come prepared and answer correctly. Let me end the session here. Thank you. Have a nice day.